Good morning, students. In this class, we will discuss about chromosomes. Chromosomes, its chemical composition, components of chromosomes, definition, function, etc., and also the types. At first, chromosomes are the highly coiled. Chromosomes are highly coiled and condensed chromatin fiber. So we can define chromosome as highly coiled and condensed chromatin fiber. So here chromatin fiber, which is made up of two substances, number one DNA and number two histone protein. It means chromosomes are made up of DNA and histone proteins. These are the two major components of chromosomes. Now chromosomes, which means colored structure, and this colored structure is called so because when it is put in the dye, it accepts dyes and it becomes colored. And therefore chromosome is known as chromosome because it is the colored substance, colored structure inside the cell. Now chromosome was first studied by Walther Fleming. Walther Fleming was the first person who studied chromosome in 1882. Since then, there are a number of discoveries are made about the chromatin fibers as well as chromatid, because chromatids are the two longitudinal sections or parts of the chromosome which are connected with each other at centromere. So we can say chromosomes are made up of two longitudinal structures. Suppose here it is a chromosome and here we can see this structure. This structure is known as, here it is known as centromere. So centromere is the spherical structure at the center of chromosome usually, and these two are the sister chromatids, and these sister chromatids are attached with each other at centromere. So centromere is the point of contact between two sister chromatids, and chromatids are the two longitudinal structures of the chromosome which are connected with each other by the centromere. Centromere is surrounded by two different structures. These are here disc-like structures, and these disc-like structures are known as what? These structures are known as kinetochore. So kinetochore is the structure. Kinetochore is the structure which is located on either side of the centromere. Actually, the spindle fiber are attached with kinetochores, which surrounds the centromere, and centromere is the point of contact between two sister chromatids. Here these are the two sister chromatids because these chromatids are the parts of a single chromosome. Here these two structures means apex of each chromatid are known as telomeres. So telomeres are the apex of apical parts of each of the chromatid. In this way, you can say this is the diploid chromosome. Diploid chromosomes means diploid chromosome, or you can say it is duplicate chromosome, because here chromosome Here it is a single chromosome, and after the formation of sister chromatids, here we can say it is duplicate chromosomes or duplicated chromosomes. So duplicated chromosome has two longitudinal structures known as chromatids, which are attached with each other at centromere. Now, chromatin is made up of two different substances, here DNA and histone proteins. DNA is present in a chromatin about 40% and histone protein about 60%. So these are the two major components, DNA and histone protein. It is a specialized protein, and the molecules of histone protein make octamer. Octamer, which is a group of 8 histone protein molecules. So 8 protein molecules, 8 histone protein molecules combine together to form octamer. And DNA thread is surrounds octamer, and these two structures made nucleosome. So nucleosome is a complex structure. It is a complex structure of DNA and octamer. Core of histone protein or group of histone protein, 8 histone protein molecule combine together to form octamer. So DNA and octamer, these are the two components which make a complex structure, and this complex structure is known as nucleosome. Actually, nucleosome is the structural unit of chromosome. So chromosome is made up of nucleosome, and nucleosome is made up of DNA and octamer. Here chromosome is also known as chromatin thread, because chromatin thread or chromatin fiber, when coiled and condensed, then it is known as chromosome. It accepts dye or color, therefore it is known as chromosome. Now, the structural unit of chromosomes are nucleosome, as you know, but the functional unit of chromosome is called genes. So genes are the structures, genes are the teeny particles or segments, specific segments of DNA which are located on the chromosome, because chromosome is made up of DNA, and the specific segments of DNA is known as genes. So genes is the functional unit, because as you know, it is the carrier of characteristic features from parents to their offsprings. So genes are the functional unit of chromosome. Now molecular structure of DNA. Here molecular structure. DNA is made up of nucleotides. It means nucleotide is the structural unit, because nucleotides combine together to form polynucleotide, and polynucleotide combines together to form DNA. So you can say structural unit of DNA is the nucleotide. nucleotide chemical composition or chemical composition or components of DNA. There are three basic or fundamental substances which combine together to form nucleotide. What are these? These are number one, phosphate, number two, nitrogenous base, and number three, pentose sugar. So these are the chemical substances: phosphate, nitrogenous bases, and pentose sugar. These are the three components of DNA. Here, suppose it is a phosphate. It is a nucleotide. It is a nucleotide. Nucleotide contains three structures. Number one, phosphate. Number two, here, pentose sugar. You can simply call it sugar. And number third, it is nitrogenous base. Nitrogenous base. So these are the three components of a single nucleotide. Now again, the second nucleotide which is combined together, here it is here again, it is complementary. Nitrogenous base, pentose sugar, and it is again phosphate. So these two are the nucleotides which are combined together by means of here hydrogen bond. So hydrogen bond connects two nitrogenous bases with each other, and in this way it is a complete structure, means two nucleotides combined together, and here pentose sugar and nitrogenous bases along with the hydrogen bond form rungs. So rungs of DNA is made up of nitrogenous base, bases, and hydrogen bond. Nitrogenous bases, these are the two nitrogenous bases, and these two nitrogenous bases are combined together by hydrogen bond, and in this way it forms a rung of DNA. And these are the two structures, phosphate and pentose sugar, which makes the strand of DNA are made up of phosphate and sugar or pentose sugar. So in this way we can compare DNA with a ladder, and here it is a double-stranded, double helical structure of DNA which was proposed by Watson and Crick. So here Watson and Crick were the persons who proposed a model of DNA which is known as double-stranded, double helical model of DNA, and he proposed this in 1953. So in 1953, Watson and Crick proposed a double helical, double-stranded DNA structure. In this structure, these are the two strands, and between these two strands, there are 5. 1, 2, 3, 4, and 5. Here again 1, 2, 3, 4, 5. So these are the 5 base pairs. These are known as base pairs. Because here 2 nitrogenous bases are combined with hydrogen bond and they form base pairs, or simply it is known as BP. So in one turn, here it is one turn of DNA and it contains 5 plus 5, 10 base pairs, and the total length of one turn is 34 angstrom. It means the distance between two base pairs are 34 angstrom divided by 10 equals to 3.4 angstrom. So between two nitrogenous bases, or you can say rungs, the distance is 3.4 angstrom. Angstrom is the unit of measuring very small distances, such as in case of DNA, because here base pairs are located at the distance of 3.4 angstrom. So here in one turn, which length is 34 angstrom, here 10 base pairs are located, and these are the two pillars which are made up of phosphate and pentose sugar. And the rungs, each rung or base pairs are made up of nitrogenous bases with hydrogen bond. So here it is, suppose helix 1, helix B, and again there are two strands, outer and inner strand in this helix, and also here inner and outer helix. Now here suppose it is 5 dash and it is 3 dash, because it starts from 5 dash and ends in 3 dash, and here it starts with 3 dash and here it ends in 4 dash. It means these are parallel to each other. Means these two strands are parallel to each other. You can say these are equidistant because the distance between these two strands are similar all the time, and therefore these are known as equidistant. They cannot intersect each other, and therefore these are parallel to each other. So these are the two strands and these are complement with each other. These are complementary in nature. Complementary. Strands are complementary. This model is known as double helical, double stranded, double helical, double stranded model of DNA. which was proposed by two scientists, Watson and Crick, in 1953. Now, there are 4 different types of nitrogenous bases may present in DNA as well as RNA. So, here types of nitrogenous bases. Types of nitrogenous bases. 4 nitrogenous bases. Number 1 is adenine, second is guanine, third is thymine, and fourth is cytosine. So here adenine and guanine, cytosine and thymine and cytosine, these are the 2 major types of nitrogenous bases found. And here adenine makes a complement with thymine and guanine makes a complement with cytosine. So here adenine and thymine are connected with double covalent bond, whereas guanine and cytosine are connected with each other with triple covalent bond. So here it is double covalent bond and here it is triple covalent bond. Adenine and thymine are the 2 complementary nitrogenous bases. They make a complement, and guanine and cytosine make again the second set of complementary nitrogenous bases. But these are connected with each other by covalent bonds, or you can say hydrogen bonds, and between adenine and thymine double covalent bond, and between guanine and cytosine triple covalent bond exists. In this way, here this one means adenine and guanine are called as purines, and here it is purines, and thymine and cytosine are called as pyrimidines. So these are the 2 major categories of nitrogenous bases. In DNA, adenine, guanine, thymine and cytosine. These are the 4 nitrogenous bases found. But in case of RNA, adenine, guanine and cytosine, these are found, but at the place of thymine, uracil is found. Uracil, which is the 5th type of nitrogenous base, uracil instead of thymine. RNA contains uracil instead of thymine, and therefore RNA is the single-stranded molecule and it is more reactive as well as unstable in nature. That's why in retroviruses such as COVID-19 virus, flu virus, etc., uracil is found, and therefore they can change itself time to time. They can make a number of strands after time to time. But due to presence of thymine in the DNA, it makes it stable and very less reactive. That's why in different types of eukaryotic organisms, variations can cause, but not suddenly. They can not react with each other or other substances to make different types of strands, and therefore the development or variations are gradual, not sudden, not repeat. So nitrogenous bases adenine, guanine, thymine and cytosine are found in DNA, whereas adenine, guanine, uracil and cytosine are found in RNA. Now, genes. What do you mean by genes? Genes are the specific segment of nucleotide or DNA. Specific segments of nucleotide or DNA are known as genes. Genes carry informations from parents to their offsprings, and therefore these are called as unit of heredity, unit of inheritance, or you can say these are the carrier of phenotypes in different organisms. So chemically genes are DNA because these are the segment of DNA. It means it is again made up of what? Different types of phosphate, nitrogenous bases and pentose sugar. So genes are located on chromosomes, and therefore chromosomes are also called as carrier of genes, vehicle of genes, and DNA is known as genetic material. Genes are the carrier of characters from parents to their offsprings, or you can say genes are the unit of heredity. Now one more thing, types of chromosomes. Types of chromosomes. There are usually 4 types of chromosomes. These are: number 1, acrocentric. These types of chromosomes are based on position of centromere in the chromosome. You can see here in this acrocentric chromosome, one chromatid is very small, whereas other one is very large. In the second, which is known as submetacentric. Here, one chromosome between 2 chromosomes, the distance or the length is different, but first is smaller than the second one. Here it is the third, and the size of chromatids are similar. Means here centromere is located at the center of chromosome, and therefore it is known as metacentric. Number 4 is telocentric. Here it is telocentric. Telocentric because the centromere is located at the apex of chromatid. So here in this chromosome, in this type of chromosome, centromere is located at the tip of the chromatid, and therefore it is known as telocentric. So usually these 4 types of chromosomes are found: acrocentric, submetacentric, metacentric and telocentric. And one more type of chromosome may be found, and it is known as SAT chromosome. SAT chromosome. Because this head-like structure is known as satellite, and it is the secondary constriction, and this one is the primary constriction. So here two constrictions are found, primary and secondary, and therefore it makes a head-like structure which is known as centromere satellite, and therefore it is known as SAT chromosome. And inside the chromosome, DNA thread is found. So it forms it is known as SAT chromosome, and this is the fifth type of chromosomes may be found in human being. Karyotype. Karyotype is the artificial arrangement of various types of chromosomes found in human cell on the basis of their shape, size. So homologous pairs of chromosomes means the set of chromosomes which are arranged or which are similar in shape and size, and therefore it is known as homologous chromosomes. So here in the karyotype, different means, as you know, in human cell 46 chromosomes are found, means 23 pairs. So all these 23 pairs of chromosomes are arranged artificially on the basis of their proper shape and size, similar shape and size. So it is artificial arrangement of chromosomes, karyotype. So in this class, we have studied about the structure of chromosomes, DNA, nucleotide also. So here one more thing, structural unit of chromosome is the nucleosome, structural unit of DNA is the nucleotide, but functional units of both chromosomes as well as DNA are the genes. So genes are actually the carriers of genetic informations from parents to their offsprings, or you can say from one generation to the next generation. And chromosomes are the carrier of genes, and therefore chromosomes are known as vehicles of genes. And DNA, whose part or specific segments are known as genes, it is known as genetic material. So in this class, we have studied about chromosome. In the next class, we will study about cell division and cell cycle. Thank you, students.